I think this is my third YIM because I attended as a mentee. Then I was organizer for one of the YIMs. The 15th one, the most difficult one that they gave me to organize, and along with partner from IIT Gandhinagar. And then this is as a mentor I'm coming to the 17th YIM. I still don't believe that I have become a mentor, but nonetheless, if they recognize, then maybe something I've done good. So to talk about, as a mentor talk, or to talk about the work I do and the challenges that I face, first thing I really wanted to say, a disclaimer, that there is nothing as an advice or a suggestion. In fact, it is just Gen Z language, BTDT, been there, done that. So I have done it before you guys are able to do it. And maybe I have the experience which will be just a sharing platform. And these are only opinions and never an advice, and very subjective and individualistic in nature. Also depends on which research or teaching domain that you belong to. Now, to actually clarify what those three words meant in my title, dodge, nudge, and push, what better could be than to give the organisms which show this behavior. Can we play the videos? So the first one is a fish actually trying to dodge. So when somebody moves towards you and you just don't pay attention and you move away, that's what is the dodge behavior. It can be quantified using an ethogram, and that's what we do it. The next one is the nudge, when you are continuously pushing the other organism to do something, maybe what you want them to do, but end up can be something different. The last one is push, and this comes in the borderline aggression actually, because you literally push and you can even displace the other organism. Why I use these three terminologies or these three behaviors? Because my model organism, this is an Amazon sailfin catfish. I'm going to tell you more about them a little more. Do show these behaviors, but also I showed these behaviors to somewhere overcome the challenges. Maybe somewhere when somebody is trying to give a free advice, I didn't want it, so I dodged that person away. When the paper is not moving, so literally push the organism, or in this case it's a human being and not an organism, and did that right. Eventually, the other organism that I work actually, can we play the video please? It's a bite and more aggressive threat. Hopefully I didn't do that. Okay, while overcoming the challenges as I became from a young faculty to a little bit mid-career faculty. But yes, you could end up being more aggressive. But then just to tell more about the passenger information and tell you the journey, I actually did a Master of Science from the Zoology Department in Calcutta University, but my thesis component was in ISC, in Center for Ecological Sciences. I worked on vigilance behavior of spotted deer against wild dog predation. Then I did my PhD again with ISC, and I worked on hormones, pheromones, and behavior in Asian elephants in different parts of India, from southern India to northeastern part of India. Then after working on elephants, I carried the hormone, pheromone, behavior physiology basket, but I then ended up working in University of Minnesota as my postdoc work on the invasive big-headed carps that are creating a lot of nuisance in the Mississippi River. Then it was a longer 6 years postdoc. I joined Ahmedabad University in 2018 as an assistant professor, and then in 2023 I got my tenure and now an associate professor. As you see here, that there were several hurdles during this phase of the journey, and one of the most difficult journeys probably so far what I have traveled, and I had many new roles to play, and I had my mentors there who helped me during in order to navigate these roles. But at the end of it, I think I'm very happy because I'm extremely passionate about ecology. For the last 25 years I've been doing ecology, and I'm happy that what I do today is extremely rewarding to myself. So to tell you the theme of that my lab works, within ecology we try to understand that if there is a population of a species A, the population, the individuals within that population are all interacting among themselves. They are also interacting with different abiotic factors within an environment, be it food, space, even nutrients, even the bad noise and the pollutants. And we try to understand these interactions on a temporal scale. These temporal scale could be season, which is a temporal scale. It could be also developmental stages of the organism. And we also try to make it even more complicated by adding another scale, which is the spatial scale, because you can have different populations of the same species interacting with each other at two different geographical area, or you could also have different populations of different species who are interacting with each other. So basically, interaction is something, biotic and abiotic interactions, which we try to understand as a part of our lab. Now, when we try to understand interactions, we understand interactions at 4 different levels: within a species, which is intra-species; interspecies, when 2 species interact with each other. For example, 2 predators in a forest, tiger and lion, how do they interact, share space, share food. We also try to understand species across an environmental gradient, because environment is drastically changing thanks to the human species, and how the organisms are adapting to this environment. Are they showing physiological adaptation, behavioral adaptation, and we try to understand how is the organism surviving in that environment. We also do community level interactions, where we put in a lot of multiple species population and their interaction with the abiotic. In our lab, we use a lot of biotic as well as abiotic readouts, and to do that, we use a bunch of tools, starting from DNA to hormones, bioinformatics, as well as a lot of chemical measurements that we do in the lab. Now, having said so, these are some of the questions that we ask, and broadly, these are the key words that we work in the field of behavioral ecology, ecophysiology, and conservation biology, trying to actually conserve the biodiversity in today's world. Now, coming to the challenges, I think these were the first few challenges in 2018 January when I landed up from Minnesota to Ahmedabad. These were the first few challenges that I had. One of the biggest challenge, and that obviously the first one I face till date, is ecology a part of biology? Is ecology quantitative? Is ecology a part of social science and humanities and may not be biology? And this is something that I continuously answer people, and my only answer to them is that, have you read the definition of biology, which says actually study of living organism? There is an organism word there, more to do than cell and molecular word actually. Then people actually tell me that if you are a field ecologist, when I joined the department, they said, okay, you are going to be a field ecologist, you work on elephants, you worked on fish, why do you need a lab? Why do we need to give you bench space and space in -80 and -20 freezer? Yes, I do need that, because I try to understand behavior, but I also try to understand the mechanistic approach to understand behavior, how or what triggers that behavior. And for that, I either have to look at their metabolism, or I have to look at their genes and or expression level, and I do need a bench lab for doing that. Then obviously there are many other, and some of it I will deal it in the few slides, like how do you balance between teaching and field work. But these were the bunch of challenges that I received. Some of it I dodged, but some I was not able to dodge. Then the next thing is obviously how I got started with, or what is the model organism that the lab works with, the themes that I discussed in the first few slides. Currently the lab works on 2 different species of fish. One is the green chromite. Green chromite is endemic to India. That means it is only found in India and a little bit in the subcontinent like Sri Lanka, but nowhere else in the globe. And this is the only representative of its family, which is Cichlidae. And then we also work on the other extreme of the paradigm, which is the Amazon sailfin catfish. I think over lunch I was just telling Anil that, we bring in a lot of pets into the country, and we don't know that when we release them, what kind of threat they will carry towards the biodiversity. Catfish is exactly like this. It's called the crocodile fish. Most individuals got it because they want a crocodile sitting in their drawing room aquarium. And then you get bored and the crocodile fish becomes bigger and bigger, and you just happen to throw the aquarium water or dump it into the drain, and there you have released an alien species, which is the potential to become an invasive in the natural biodiversity. One of the hurdles that I faced as a young PI when I joined Ahmedabad University was setting up a fish housing facility. The department already had a zebrafish facility because there were my colleagues who work on zebrafish. But I was trying to set up a facility which is more natural to their environment. So I really didn't want RO water, chemical treated water. I really wanted a mesocosm kind of setup. I also wanted to watch their behavior from overhead. So I wanted a kind of floor arena so that I can remotely watch their behavior and that camera can talk to the laptop or to the computer sitting somewhere. So in order to overcome that challenge, what I did is while I was at University of Minnesota, I planned few research questions for my fish. I was very definite that I wanted to work on fish when I come to India. So I planned a few research questions and objective. I also had decided that I will want to work on green chromite because I did a literature review and I knew that it will be very interesting to ask some of these questions to green chromite. I even wrote draft proposal. But the other thing that I did is I also took detailed notes on plumbing or even the housing facility that was there in my postdoc supervisor's lab because he was also working on a wild species, the big-headed carp. So that really helped me when I actually came to India because I sat with the plumber, I sat with the electrician there and whatever they say that madam is tarike or in this way if we make it, it's more going to be energy efficient. I said no, listen to me, I know I want to do the experiment so do it my way or the highway kind of. So that helped me. Also the grant drafts that I wrote while in the last year of postdoc that helped me to immediately submit. That time it was still DST SARB and I got my first CRG large grant by actually on fish proposal. I also, when I joined, I only did not depend on PhD students because being a new lab, being a new PI who has done PhD 7 years back or 6 years back, nobody will remember you. So I actually vested all my interest in the undergrads. Being a university, we had a very good undergrad population and several undergrads actually came to the lab because there was something new. I was the first ecologist to be hired on campus. Several undergrads came to the lab and we did some really beautiful work. They also got a lot of co-authors in the publication. I also could standardize, optimize and did some wonderful experiments which are very much curiosity driven. So the fish lab or the housing that I was given to set up the fish lab actually was a bioinformatics or computer lab. Somewhere I pushed that instructor away and then was eventually getting the first fish tank and then the whole setup kind of looks like this. This is what the overhead, this took the largest amount of effort to set it up. We use an overhead camera to watch the fish from beneath us in these tanks. So this was one challenge, but while I was doing the fish work, at the back of my mind, I also wanted to work something which is a wildlife species, more to do with the larger vertebrate because elephant I didn't want to work, that was a very conscious decision. My PhD supervisor is a big shot in elephant and I really didn't want to be a seedling under the shadow of a large tree. So that's the reason I very consciously took the decision that I don't want to work on elephants and I want to work on a wildlife vertebrate species. But I was not sure what I want to work on. So, but fish work event was much, dates back and it was already started. We asked some very basic questions in the first years, like how do they form groups, do they show shoaling behavior, how are they identifying their shoaling partners, how is that driven by their preferences, are they using vision, are they doing olfaction or tactile, and then eventually now, and as you can see the fish is also getting sexually mature because initially you are setting up a wild population, there will be a lot of die-outs and population crash, but now we have a very good sustained sexually mature population and across developmental stages, and around this time you can ask complex questions. So all the young YIs there, I would like to say that yes, you can actually ask very complex questions, but you may have to wait till your lab evolves or your infrastructure evolves and take you to that stage. And now we are kind of asking very complex question that how the opsin or the visual pigment gets altered when a fish moves from a fresh water to a turbid water, from daylight to the places where there is hardly any light because pollution would have killed or would have obscured the light in these environments. Similarly, with the next fish model, we again asked from simple questions to more complex. We also first did laboratory-based work on the fish, then moved into fields where we have set up large ponds mimicking the natural ecosystem in order to understand that how the catfish, which is an invasive because it came from Amazon, is impacting the native community. We collaborated with West Bengal and Orissa Fisheries Department because it was impacting the economically important rohu or the freshwater fish, which gives the country a lot of economic benefits because it's highly marketable. And then we also looked at to develop a molecular tool to understand the measure the catfish in water using the eDNA methods. But that were more complex molecular tools where towards the end and the initial start was more simpler question, and which also gave us publication, which also gave the undergrads publication. There are several undergrads in these papers, which also help us build the lab and get going. I think what Siddesh was trying to say that you have to be consistent. CNS is good to publish, but keep publishing in society journals to have the name out there. So then, if I'm doing it, what I will come back to, or what I mentioned just before, is that what is the wildlife species? And for the fish work, I had to travel everywhere to get the fish to population, and it was continuously in my mind that what is the wildlife species I will work on. And while being in Gujarat, when I was traveling for all these fish work, I came across this freshwater, cold-blooded animal or a large vertebrate, the freshwater mugger crocodiles or the marsh crocodiles. And I started doing a literature review. I look at the papers, I talk to people, my own peers and mentors, that is there any work on the ecology, behavior and physiology of muggers? And there was not much. In fact, most of the work was way back 1970, 1975, and most of it by Romulus Whittaker, who had set up the Madras Crocodile Bank Trust in Chennai. So I went there, that's Zai Whittaker, Nikhil Whittaker, and Soham, who was leading an NAJA NGO and partly working on turtles and reptiles in Gujarat. I spoke to them and I got to know more about the crocodile biology, got myself a learned member of this reptile community because I'd never worked on reptiles before, in order to actually think about the relevant questions, objectives, so that I can function or probably start looking into the adaptations of the crocodiles, because these crocodiles live in multiple different habitats, from river ecosystem to pond to even desert ecosystems in Kutch, and they must be doing a bunch of things in order to adapt to these diverse environment, which actually triggered the interest in me. But by then it was 2020, and we all know what happened in 2020. We were all sent back from our labs and fields back into our houses and locked up because COVID hit us. Now, while sitting at home, and I had one master's thesis student, one PhD student who just got started on mugger crocodile, they were very stressed, okay, and they stressed me even more that what is going to happen now, how are we going, 1 year is getting lost, and how are we going to progress? So while, I have 10 minutes, okay. While doing all this, we thought about it that, while being in the part of the wildlife management or wildlife biology, I know that you need to track, tag, or put radio collars in order to understand the individual level physiology, behavior in animals actually. How about if you can develop a non-invasive method in order to actually individually identify crocodiles, because that is going to feed in very well with our kind of questions we are asking, because maybe a dominant individual is acting very differently than a subordinate one, a sexually mature is acting very differently. So how about getting some individual level data? But how do we go about in making our non-invasive method? So what we did, can we have the video? Is this is just we work a lot with drones, and this is just a drone footage from Kutch area. the crocodiles have something which is called the bony plates on their dorsal side, which is cute. If you can like go towards that end, maybe. I thought this video was smaller. Or maybe it will come. So that's what. So these patterns that you see on their back are bony osteodermal plates actually, and like our thumbprint, these is unique to an individual. So in collaboration with Mehul, because I was in university, me and Mehul for the next 1 year did not meet each other, although we were in the same university, and we had a paper before we met actually in person. We used AI ML, like deep learning tool, in order to develop an algorithm using drone-based images in order to identify crocodiles individually. Right now for Gujarat, we have data for 220 individuals who are all individually ID'd in our library, and we are trying to now we got a grant from Island Foundation, from IUCN, as well as from CSR Aspire, and we are trying to actually develop into an app so that as you do the field work, I will get to know, is it my individual 1 or 143 or 240, whatever. So these are multiple different things, the crocodile work we are doing. We work in these places, Kalchera, Anand, Vadodara. We are also doing nighttime monitoring of behaviors and trying to take their skin temperature using thermal camera, because how does a large cold-blooded vertebrate adapt to this climate change paradigm of the climate change environment. And I also want to tell everybody out there, it's very important to actually find out the time to write popular articles. It not only helps you to get philanthropic funding, but it also actually helps you to talk to the undergrads who may not be that familiar to the technical jargon, but will probably understand an article in Mongabay or Down to Earth or even in a newspaper. And we keep doing that periodically for a peer review article. We always try to actually have a counterpart as a popular article. Now, teaching, I think it was altogether a different ball game, but I love teaching. Because I was the first ecology faculty on campus, I had to develop the first few ecology courses. Before I joined, it was ecology courses was treated, only one course was there, that also was taught by an adjunct genetics person, and it was only there to fulfill the UGRC requirement actually. So it was a huge struggle to actually include ecology in the core course, but my students and their feedback really helped me to actually build the ecology, and now we have 6 to 7 courses, both core and electives, which we run as through the ecology. Through my students also, I could actually get more questions, go with them out in the field and get exposed, because right there is the organism of the community sitting, and you could be asking very curiosity-driven questions, which will be can be converted into a scientific project. Besides that, there was a lot of tweaking in the administrative policy. For example, how do you do field work, because you have to teach here. And many people, when I was trying to work on muggers, told you should work on a backyard species, because you have to balance teaching, admin, as well as field work. There's nothing wrong in working in backyard species, but you have to have the calling. And my calling was more for the crocodiles and the wildlife. But I kind of tweaked the attendance policy with the help of the dean then, in order to say that field work is like attending a conference or even more than that, so give an authorized absence. Gave a lot of different kinds of tutorials or research talk in different departments, even in front of the dean and provost, to actually help and seek the support towards the ecology. One thing that I would like to also bring in here, that one major change in the policy that I had to bring in is the POSH policy, prevention of sexual harassment. We send our students to remote areas in the deepest forest or deepest river, like some river, and the POSH policy was only applicable to the campus or to the larger campus. It was never applicable to my students who was working deep inside the forest or in some Vishwamitri river in Gujarat, and I had to tweak the policy. Several I took help from different other disciplines because even anthropology, geology, humanities and social sciences faculty also have to do field work. That's how I built in guidelines for POSH. And then several ecologists now have come together, 47 of us across India, and we have developed a POSH policy for conservation and ecologists particularly, that how the policy or the guidelines should dictate your field work or your work. So having said that, doing conferences are important. I did many different conferences and there's a huge support system. Nobody can work without support. There is a lot of support from different things, but I think the majority support is the students. Lab in 2018 and this is the lab in December 2024. They are the Polans in the system. They will carry your message to different other institutes and ecosystem and help you spread the word. So that's how I depended on my students for that. I have a huge help and thanks I want to say to all the field assistants who are tribal people and local villagers who are part and parcel. Without them, we are almost dysfunctional in a forest or in an ecosystem. So huge thanks to them as well as the forest department. These are some other key takeaways which I can talk, but the main thing is that please be passionate, breathe. There is nothing called success. I think we just have to stay afloat. And with that, I will end and I can take any questions.